Hello, everyone. My name is Ning Dai. I'm a associate professor at the Department of Civil Structural and Environmental Engineering at the University of Buffalo. It's a great honor for me to give the Burges lecture today and for visiting the UWC department for my sabbatical this fall. What you're hearing right now is a re-recording of my seminar in November because we had encountered some technical difficulty in the audio setup. Okay, so let's dive right in. Before I give the, introduce my projects that I will cover in today's seminar, I want to give you first an overview of what we do in my research group. So a lot of the effort focused on the disinfection byproducts and membrane processes in the context of water treatment and reuse. So if you're not familiar with disinfection byproducts, I will introduce them in a few slides. But overall, a lot of our effort are focusing on the molecular scale mechanisms with the hope and intention to predict and understand the behavior and performance of systems. For example, in the disinfection byproduct aspect, we are interested in, studying the formation mechanisms, the chemical pathways, with the intention that this knowledge will help us to predict or come up with control strategies of these compounds or watershed management strategies to prevent their formation in the first place in our water systems. And on the membrane performance front, we are interested, for example, how small organic molecules, many representing contaminants of emerging concern, such as disinfection byproducts, how these compounds transport through the polymer membranes. On the other hand, when we have these polymer membranes, how do they degrade over time when we put oxidant or disinfectant into the system? What fascinates me a lot about engineering systems is that in our design, in our daily work, we are trying to we're trying to balance many different competing objectives. And it's certainly so in environmental engineering, right? Because we want to maximize the system performance without inducing unintended consequences. And that's what we do. So today's talk, I will cover some example projects under these two categories. Outside of what I have on the screen right now, my group also does a lot of sunlight photochemistry on environmental surfaces. And I don't have time to go into those today, but I would love to have an opportunity to, exchange ideas with you on those when time happens. So, all right. So the next thing I would like to do is to acknowledge the students who contributed to the work that I'm presenting today. So Zach and Tashfia are two senior graduate students in the group who contributed the work I'm presenting today. And specifically, I would also like to acknowledge Jala Xu, a former student who contributed the work that Zach's current work is building on. And Dr. Putumanalam is my collaborator from the materials department at the University of Buffalo. And I am also very appreciative of my research group. It's great fun working with them and also the funding agencies. So let's go into the first project. That is a story about how we explore the intermoiety as a nitrogenous DBP precursor. So let's talk about disinfection byproducts. So what are they? We know disinfection, which is important, right? Because they inactivate pathogens such that we don't get a waterborne disease from drinking water. And disinfection is actually one of the greatest, public health achievements in the 20th century, significantly improved, life expectancy in the public. However, the disinfectant that we use can also react with various water constituents. Many of themselves are not harmful to convert convert these constituents to form disinfection byproducts. Most of the disinfection byproducts are present in low concentrations. They're nanogram to microgram per liter level. However, they represent a chronic exposure burden. And some disinfection byproducts are indeed probably human carcinogens. So that brings us to the first, challenge, right? So how do we balance the microbial or biological safety of the water that's from pathogens and the chemical safety related to disinfection byproducts? So currently, the US EPA regulates 11 disinfection byproducts, and here are some examples of them, so like the trihalomethanes and the haloacetic acids. What maybe in the last few decades people realize is that we have different new water constituents in our waters that lead to the formation of emerging disinfection byproducts. I would like to, qualify these new and emerging. Some of them are genuinely new because we are, putting out new synthetic chemicals into the environment every year, right? A lot of them, and some of them can serve as precursors to the disinfection byproducts. At the same time, because the analytical technique is developing, so sometimes we are detecting water constituents, detecting disinfection byproducts that we didn't know before, and some of them do represent a great, health risk. And among this change in water quality, what researchers really starting to realize are those nitrogenous DBPs, means that the compounds containing nitrogen, for example, here the nitrite group, the N-nitroso group, or the nitro group here, these compounds represent much greater toxicity than the ones that are currently regulated by the EPA. And my research group is really interested in one group of these nitrogenous DBPs, namely the haloacetylnitriles. So we see a lot of HANS abbreviation, and those are the haloacetylnitriles. Researchers found, to the knowledge that we know, among the known DBPs, haloacetylnitriles represent a major toxicity driver. So for example, if we look at this pie chart, on the left-hand side is the, I forgot a heading, is the mass concentration of all the different group of DBPs. So haloacetylnitriles are here represented in green. On the right-hand side is the toxicity, the calculated toxicity of the overall contributed by all of the DBPs. And you can see here, the halonitriles represented about 45% of the total toxicity, along with some other group of nitrogenous DBPs. And on top of that, people also know that brominated species are generally more toxic than the chlorinated species. For example, this is a relative toxicity on a log scale plot. And we have here the haloacetylnitrile that have two chlorine. It has two chlorine, like here. And here we have one bromine, one chlorine, and then this one has two bromine. So you can see their toxicity really increases at the log scale. And by the way, if you look at the scale here, this is the relative toxicity relative to chloroform, one of the regulated DBP. So we are seeing, haloacetylnitriles are like thousands of times more toxic. So to study haloacetylnitriles, we want to find out how they form in the first place, right? So what are the precursors? So the classic theory is called a amino acid decarboxylation pathway. So basically, if you find that way too much chemistry, just think about where does the nitrogen in the haloacetylnitrile come from? And the very popular or very well-known pathway is to come from the nitrogen in the free amino acid. So this is an amino acids, and this is the amine group. So basically, the idea is that upon chlorination, undergo a series, processes, they will form an imine, so the CN double bond, which undergo further reaction to form the nitrile group. Also researchers have shown that, if you have an electron withdrawing group attached to the beta carbon, such as, the compounds shown here, that you will have a high yield of haloacetylnitrile. So these are some examples of the haloacetylnitriles. However, if you really think about this, amino acids, most of them in the environment are actually bound in peptide bonds, like in small peptides or in the whole protein, right? So they are in this amide bond. And it's very well known that the reactivity of the amide bond with chlorine is much, much lower than amine. Additionally, our previous work showed that there's something interesting about the different, the brominated versus chlorinated haloacetylnitrile precursors. So this is a paper that we did. Basically, what we did is that we took wastewater effluent from different treatment plants. We irradiated them under sunlight, and then we watch how the precursor concentration change over time. So if you think about the y-axis as the precursor concentration, the blue is the precursor of the chlorinated halocyanitrile. The red is for the brominated, the bromochloroacylnitrile, so have some bromine in it. What we realized, wait, interesting, the brominated halocyanitrile precursor was degraded by sunlight faster. So that implies, wait, we may have a different pool of precursors, right? So that brought us to think about, well, if you have an amino acid themselves, they're not really sunlight reactive unless you have these kind of aromatic functionalities. So it prompts us to think maybe there are other halocyanitrile precursors out there. They're aromatic, such that they absorb sunlight and they have nitrogens. So that's why we started with the indole moiety that's in the tryptophan. So tryptophan is a known relatively high yield halocyanitrile precursor. However, most of the effort focused on the nitrogen here without looking at this nitrogen. And the reason why we are interested in the indole group is that, as we said, tryptophan has this aromatic nitrogen containing functional group. And also our research, as well as many others, have shown that the fluorescence signal of tryptophan-like moieties correlate with the halocyanitrile formation potential. So this is an excitation emission fluorescence matrix. So basically the idea is that in this 2D matrix, the region, the signals here represent constituents that are like tryptophan. Not necessarily tryptophan themselves, but like them. And if you plot the tryptophan fluorescence, tryptophan-like moiety, the fluorescence intensity with the formation potential of the HANs, they do have a very good correlation. So, and then we think about, wait, which is the moiety that fluoresces? It's definitely not this part, it's this part, right? So, and then lastly, tryptophan is sunlight reactive. And that reactive component, again, is on this indole moiety. So all of this brought us to think about, let's look at what is happening in the indole moiety. So what we did is that we took two compounds. Actually, we took more, but here in the results, we should want to show fewer, just make the story a little bit simpler. So we took tryptophan that has the nitrogen and the 3-indopropionic acid that is very similar to the tryptophan, except that it doesn't have this free amino nitrogen here. And then we look at we chlorinate and brominate them and see how the HAN forms. So on the left figure, you have the chlorine, the dichloroacylnitrile, so chlorinated experimental results. And on the right-hand side is the bromination experimental results. And on the x-axis, the time. The red symbol represents the results from the tryptophan. So what we see here is that tryptophan indeed forms a lot of halocyanitrile. And in this time profile, it has a very interesting initial rapid increase. And the blue curve representing indopropionic acid, IPA, it does form halocyanitrile steadily. And at the 24 hours, the halocyanitrile formed from IPA represents about half to 85% of the halocyanitrile formed by tryptophan. So we can see this percentage is greater in the bromination case. I would also like to note that these are the experimental conditions conducted at the same halogen to nitrogen ratio. So that means that actually there is twice as much free chlorine and free bromine in the tryptophan case. So this results really confirm that the indole moiety is forming HAN substantially. So later we want to do a even further line of evidence. So we took this compound that has a N15 labeled amino group, so it's a tryptophan, but the indole part is still N14, but the amino part is N15. So then we look at the halocyanitrile formed from this compound using AGC MS instrument. So if we look at here, the red part of the bar represent the halocyanitrile from the amine part. The dark gray is from the indole part, which is the N14 halocyanitrile. So we see here, very similar trend as the previous time profile, right? So the indole moiety contributed to the total halocyanitrile formation from the tryptophan. And in the chlorination case, somewhere in the 30 to 40% at the end, at the 40 to 50% formation case. So again, slightly higher than in the bromination case. So both of this line of evidence really concluded very conclusively saying, okay, the indole moiety, even though nobody have talked about them before, they actually contribute to the formation of halocyanuriles. Then we want to know, well, how does this component actually form the halocyanitrile? So we used the compound without the free amino nitrogen. We proposed some pathways. As in Zach, based on some literature review, proposed two pathways. The first one, he named it as indolanine pathway. It's actually based on some studies from the 70s on the chlorination of indole free or substituted indoles. So basically the idea is that you form this intermediates, have a halogen substitution here that it's called indolanine. That's how the pathway is named after. And a second pathway is what we call the aniline pathway. What it represents is that upon halogenation, there is a ring opening on this indole functional group that forms a aniline-like intermediate. And this is a degradation pathway drawing the analogy on studies that have been conducted on the tryptophan photooxidation or tryptophan oxidation. So the way we kind of probe this mechanisms was using an LC Orbitrap high resolution mass spectrometry. We conducted halogenation experiments using this compound, the precursor, the 3-indopropionic acid. We use a lower molar excess of halogen such that we can, capture the intermediates. We don't want it to go all the way to the halocyanitrile. And also we freeze dry the samples and reconstitute in water to concentrate them. And using the LC Orbitrap. So I know many of you in the department actually do high resolution mass spec. So if you are unfamiliar with it, basically you can think about this as a tool, a new mass spec tool that, because of the high accuracy of the mass, such that we are much more confident in the molecular formula that it generates, such that we can more confidently say, okay, this intermediate is likely to be that one. So we, using the LC Orbitrap in 2 molds, we first do a full scan, and then it's kind of similar to a suspect screening workflow based on our proposed pathways. And then the second one is once we see this potential intermediates, we further fragmented them such that for further structural elucidation. All right, so basically, put it in a more simple way, basically we detected features that matching each of the pathways. We detected 6 features matching the indolanine pathway. However, 2 of the features are actually potential isomers of the aniline pathways. So hold that thought. And then if we look at the aniline pathway, we detected a lot more of the features that are matching the molecular formulas. If I didn't explain it quite clearly, but basically the idea is that what Zach did is that, when we draw this chemical structure, each intermediate that have different possibilities, right? Different halogenation, hydroxylation. So he come up with this like really long list of the candidates. So he does the matching of these features that we see to that candidate list. All right, so we, based on this, come back to like in the aniline pathway, we definitely see more potential matching of the features. So Zach look back and say, well, how do we communicate confidence of this, matching? And of course, everybody goes with this very well-cited Szymanski scale as kind of a standard of the community. However, he also realized that unlike some of the work that are using high resolution mass spec to identify small molecules in environment matrix and or commercial products, here we are really starting with known compounds. So we are kind of almost always starting with a known structure to begin with, and then kind of build up. So in this level 3, he further kind of gave a final resolution of what those would be. For example, you can have a level that the probable structure is more tentative, because we cannot, based on the site, based on the fragmentation, we have a probable structure. However, we cannot decide where the exact site of the halogenation is, and that is usually a challenge in DPP kind of intermediate identification, because the halogen does leave preferentially during fragmentation. So, but basically, this is what Zach wanted to communicate, the confidence. But let's actually see a little bit more what the story is about here, is that we used fragmentation to kind of identify or to give better information about the structure, such we can differentiate the potential isomers that would happen in both pathways. So for example, if you remember earlier, I said in the indonolin pathway, we identified 6 features, but 2 of them actually were possibly an isomer or actually a compound in the aniline pathway. So Zach, what he did is that he find these features, he asked the MS to fragment it and look at the fragments. So if you look at the fragments, for example, based on this fragments, it's very clearly that this compound, that the feature that we see is not an intermediate from the indolin pathway, but rather one from the aniline pathway. So in conclusion, is that we use all of these kind of mass spec tools to probe and investigate a system. We overall identify many more candidate intermediates that are from the aniline pathway. So the aniline pathways is our, go-to. So what is, that's great. Zach is writing the manuscript, hopefully to be published soon. I hope to submit it soon. And then the next question is that, so what next? So what really intrigued us in this process is the identification of the aniline intermediates, because this pathway can really help us to think about a broader suite of compounds. So they are very common in synthetic chemicals, say, for example, pharmaceuticals, pesticides. In fact, recently that there was a 2022 paper showed that sulfamethoxazone, a pharmaceutical compound, actually has a pretty high halocyanitrile yields, and we highly suspect that is because of this aniline moiety. And aniline also have a lot of derived structures such as amide, arylurea, tertiary, secondary, and tertiary amines. On top of that is in a collaboration between my group and Dr. Jindra Ju's group, who is a water quality modeler, we have noticed that at least in the Illinois River watershed, there was not a lot of the non-urban land use can potentially contribute halocyanitrile precursors. So that made us to think about, what else? What are the other synthetic chemicals out there that come from, non-point sources, for example, agricultural runoff. And so Zach, very quickly went to the lab, took a series of aniline compounds, and with all different substitutions, for example, just aniline itself, or substituted a 2 precision, 3 precision, 4 precision with different substituents, and then did a quick assessment to look at the halocyanitrile formation potential. Again, on the left is the chlorination, right is bromination. We plot the tryptophan as a reference. So here we see is that most of the aniline have lower, but some have comparable formation of halocyanitrile as the tryptophan. And in the bromination case, while many of the aniline have actually much greater formation of dibromo acetonitrile formation than tryptophan. So really made us think that maybe this is a really important precursor. So from there, that's our next project, and hopefully in collaboration with Ed's group, so that we can further investigate the system. All right, so that's enough of DBPs in the last bit of time. So I would like to give a quick overview of or introduction of the projects that we have been doing on the oxidative degradation of polyamide membranes under conditions, exposure to prussic acid, free chlorine, and chloramine. So this, again, is Tashfish's work in collaboration with Dr. Putmanallam at UB. So let's talk about disinfection in membrane-based water treatment system and how it's slightly different in terms of the intention compared to a conventional drinking water treatment system. So this, what I'm presenting here is what we call a full advanced treatment train to convert secondary wastewater effluents all the way to potable quality for indirect or even direct potable reuse. So this wastewater usually, the secondary effluent usually go through microfiltration, reverse osmosis, and advanced oxidation. So disinfectant is typically added, oftentimes used like, say, for example, chloramine for membrane fouling control. So we know that you may notice that we are using a relatively weak oxidant here. And the reason is because strong disinfectants, such as free chlorine, actually degrades the polyamide RO membrane. So the industrial standard for RO membrane is this thin film composite that has a very thin layer of polyamide active layer. On top of that is the selective barrier for the membrane, the most important component, and free chlorine degrades it. At the same time, chloramine and or any kind of disinfectant can react with wastewater, effluent, organic matter to form disinfection byproducts. In this particular case, for example, NDMA and nitrosyl dimethylamine is actually formed pretty prevalently between chloramine and wastewater, effluent, organic matter. And some of these small molecules, very highly toxic, both of the ones that are listed here are very toxic, and the small neutral DPPs also very poorly rejected by the RO. And in fact, that's one of the reasons, like NDMA is one of the reasons that we always use a UV-based advanced oxidation such that we can remove it. But some other halogenated nitrogenous DPPs are not so well removed actually by the AOP. So they represent some potential risk and concern. So based on this destruction, you can see that disinfection for the membrane system is balancing different parameters. So on one hand, we need fouling control, and that's why we added it in the first place. At the same time, we don't want it to form a lot of DPPs, and at the same time, we want to make sure that the membrane remains intact so that it can continue to do the continuous performance. And that's what prompted us to start looking at prussic acid as an alternative disinfectant. From a microbial inactivation perspective, we conducted some bench scale and also pilot scale experiments, and along with many others in the field. PAA disinfection performance is close to, not quite exceeding and completely on par, but very close to free chlorine, which means that it's definitely much, much better than chloramine. At the same time, most of the studies have reported that PA has a relatively low formation of DVPs, especially if your water doesn't have high halide to begin with. So this is the structure of prussic acid. It's a organic peroxide, basically. So the knowledge gap here is that, with this three-way balance, we know the fouling control related to disinfection strengths, DVP control. So now what is the polyamide compatibility of PA? That's the remaining question. So in an earlier collaboration with Dr. Boyaxiong at the University of Minnesota, we used commercial polyamide membranes and compared its membrane compatibility with free chlorine. So here's a figure showing the pure water flux. So basically, if you look at the figures, that whatever the curve that you see, if it's closer to this gray dashed line representing a pristine membrane, that means whatever that we are adding into the water does not harm the membrane. So you can here see the curve for PAA in green and the red for chlorine. So we can see overall the performance of PAA exposed membranes is much closer to the pristine membrane compared to the free chlorine exposed membranes. However, we obviously want to caution us to think about, well, these preliminary experiments definitely suggest that PA led to relatively small changes in the membrane performance. We want to kind of dive further. How is that membrane performance change versus the change in the polymer structure and property at the molecular level? Because the truth is that the latter is more important in terms of predicting a long-term membrane stability. So that's when we move into a new kind of platform of probing this mechanism. And that's where Dr. Porter-Manalum's expertise came into place. And then the technique that we're using is called a quartz crystal microbalance with dissipation. Basically, it's a very sensitive balance detecting small mass changes on the surface. It relies on the piezoelectric materials that vibrates under a certain electric voltage. So later you will see a lot of figure that looks like that. So in the axis, you have time. On the y-axis, you have the change in the frequency of the vibration. You have a change in the dissipation of the energy. So usually, if you have a solution pass through on top of a quartz crystal microbalance, like the quartz crystal, or you can call it the sensor, if whatever is the solution does not interact with the surface, or there's nothing in the solution, like a DI water, you have no change in the frequency. So if you have something in the solution that interacts with the surface, then means mass is gaining on the surface, then the frequency will drop. And the drop in the frequency can be related to the mass change on the surface. And sometimes you have a rinse that you will remove mass from the surface, and the frequency change will be positive, as in frequency will increase. At the same time, you can have a change in the dissipation. So basically, overall, the idea is that when frequency drops, that means mass accumulates on the surface. When dissipation increases, that means that the attached mass becomes more viscoelastic. I think I'll briefly touch on the dissipation, so we'll save it into a later slide. I'll dive a little bit more. So basically, overall, the project design, we used two different types of ways to synthesize the polymer, and either by molecular layer by layer, or we use a authentic, commercial membrane, isolate the polyamide layer and transfer it to the sensor. We probed the system using QCMD by exposing the polyamide films to PA solutions, free chlorine chloramine, and then we also supplemented with other characterization techniques. So here, I think for time's sake, so I'll probably most focus on results from these experiments. So this is a graph showing how polyamide film changes upon exposure to PAA. So first, you have a period of gaining the mass on the surface, and this is what we call the sorption phase. And then later, you have a degradation phase. The frequency increases very substantially, and that's where the PAA start to degrade the polyamide and losing mass from the surface. So from here, we know that obviously we're using a very, very high concentration of PAA, but we would say it does lead to a polyamide degradation. And about 20% of the polyamide, there was a 20% of total polyamide mass was lost at the degradation equilibrium here. So this is an AFM image of the final quartz crystal. What we can see here is that the mass, the polyamide loss, is due to this formation of pit. So some part of the polymer just went away. So that it's not a smooth AFM polyamide surface anymore. And in the dissipation, we also see a substantial increase in during the sorption, especially the later sorption phase and the degradation phase. And that implies that, PA incorporated into the polymer and also degraded the polymer such that the polyamide film becomes less rigid. In the QCM data, kinetic modeling of the data can tell us a lot about the fundamental kind of microscopic mechanism. So what we did here is the sorption phase. So I want to highlight we're modeling the kinetics in the sorption phase of the PAA polyamide interaction. But we converted the frequency change to mass now. So that's why it's going upwards. So what we see here consistently in all 8 replicate experiments, we see a 2-stage sorption behavior. Stage 1 is a initial that fits better a pseudo first order model that can imply that the molecules are mostly absorbed on the surface of the polyamide. And there is a stage 2 kinetics that fits the Boltzmann sigmoidal model that often used to describe competing sorption degradation reactions, such as the enzymatic reaction. So that is what we believe, that the PA start diffuse into the polyamide network, and then also start oxidizing the rings. So how does PA compare with free chlorine? So the bottom figure is what you have already seen, the PA results, and then the top figure is the free chlorine results. So we can see here a very rapid change in the frequency, such that we need to kind of zoom in. So upon exposure to free chlorine, you do see an initial dip in the frequency, but it really is a very short amount of time, and very immediately you see the frequency increases. That means that the polyamides start to degrade. So overall, free chlorine induces much faster polyamide degradation. That PAA is like minutes to hours, like versus hours in time scale, which confirms our previous observation, right? PAA is much more compatible with polyamide. We also note that here we're doing experiments with actually 10 times lower concentration of free chlorine, because otherwise we just basically cannot resolve the system. And at the end, with this even lower concentration of free chlorine, about half of the polyamide film loss, compared to 20% by PA. So overall, these are promising results, as what we can say is that both at the molecular and membrane performance, just two ends, we showed that PAA is more compatible than free chlorine with polyamide. But we're also very clearly about this, is that chlorine, we use it only because it's a well-studied disinfectant. But the truth is that in the field, people don't use chlorine because it's terrible. So we're showing something that is better, potentially a lot better than something that is terrible, but it doesn't mean that PA is good, right? So we're moving into the next phase of the project, really is comparing PAA versus chloramine. And some preliminary results suggest that actually PA is potentially even better than chloramine in terms of membrane compatibility. But I want to highlight some interesting findings that when we look at the chloramine results here. As before, upon exposure to chloramine, we see a frequency change, like drop. So monochloramine is absorbed on the polyamide membrane. But if you look at the results here, this is the different frequency that we detected. A different kind of overtone. I think that I missed that introduction. But basically, think about this, is that the QCM instrument not only can detect mass change, but it can also probe the gradient of the mass along the depth direction. And what we see here is that the chloramine interacts with polyamide. The different frequency change across different overtones that suggest that there's a depth-dependent processes. And our current hypothesis is that this depth-dependent processes has a surface absorption, diffusion, and reaction, multiple components. So our current ongoing work is trying to use additional technique, for example, mechanics of the membrane under the atomic force microscopy, force spectrometry, and nanomechanics to decouple these two processes. But work is ongoing, so hopefully we'll have something to report next year. All right, so as a summary, this is the idea of the oxidative degradation of polyamide membrane. We try to use a new platform to study the process, and I learned a lot about these systems from my collaborator. So it is a fun process. So with that, that's all of the presentation I have. So I know it's an online recording, so I couldn't take any questions, but I hope you enjoy the work and hope you continue to follow what we do.